This conference will now be recorded.
Hello everyone. Am I audible to everyone?
Yes, I can hear your voice. Yeah.
Yes, thank you. So let's wait for a few more minutes to remaining speakers to join. We'll start within 5 minutes. Well, since I hope everyone is here and we have a lot to cover, we really should begin today's webinar, and I really appreciate you all for attending today's virtual conference. A very good morning, good afternoon, or evening to one and all present here virtually with me today. Let me introduce myself. My name is Elena Jones, Program Manager and Conferences Secretariat at Coalesce Research Group LLC, and I will be available for any of your queries or questions throughout the webinar to help you technically and organize the webinar successfully. Finally, I would like to thank you all the attendees and participants of today's virtual conference from 5th International Webinar on Plant Science and Molecular Biology, and hope you enjoyed the webinar and gained some knowledge from the sessions today. Also, we hope your participation will contribute to new research findings and that will lead to new collaborations and research initiatives. Before we start with the session, I guess some of you will be new to GoToMeetings app. Let me just explain you the important tools required for the presentation. On the GoToMeetings app web page, you can find your 4 icons at the bottom center: mic, camera, screen, and end button. Mic button. Click on the to mute and unmute. I request you all to stay muted while our speakers are presenting their research work so that it doesn't cause any disturbance to them. Camera button. You can use this button to share or unshare the web camera. If you have the option to share the webcam, you can do it, and it is completely your choice. Screen button. You can use this button to share your screen. This is the main part. During your presentation, after I call your name, from my side, I will be making you the presenter, and only I will give the permissions to share the screen. Then only you will be able to share the screen. So once I make you the presenter, you can click on the screen button and you will find a pop-up window to share your screen. And end button. This is the 4th icon where you can use this to leave the session. On the top center, you can use the options to change your display settings. At the top right corner, you will have 3 icons: people, chat, and settings. In the chat option, you can chat with me or with any other participants. For the settings, you can use this button to check for the settings of your speakers and microphone. So as mentioned, kindly remain muted during the presentation so that the presenters don't get any disturbed. And for any questions, you can ask the questions or you can send your questions in the message box too. Well, let's not take much of your time. Finally, I would like to request you to please give us a brief introduction about yourself before starting with the session so that we can know about you virtually. And let's move on to our sessions. The first presenter is Dr. Chik Khong Yap from University Putra Malaysia with the title of Enzymatic Antioxidative Activities as Biomarkers of Potentially Toxic Metals Exposures in Centellastic, a medicinal plant. Hello, Dr. Chik Khong Yap. Are you ready with your presentation?
Yes, I'm ready. Yes.
Okay, so now I'm going to make you the presenter. Please click on the screen button to share your screen.
Can you help me to? I already email you my slide. I think you received my email.
Yes, I have received your email, but
Yeah.
do you get any pop-up window?
Okay. So I click screen. Am I right?
Yes. Yes, we can able to see your screen.
Can you see my
Okay.
screen? Okay.
Yes, doctor. Please continue with your talk.
Sorry.
Yes.
Go EE.
Yes, perfect.
All right. Thank you. Okay. Now a very good day to ladies and gentlemen. My name is Yap. I'm a full professor in my University Putra, Malaysia. So the title of my talk today is about enzymatic antioxidative activities as biomarkers of potential toxic chemicals or heavy metals exposure in a popular vegetable plant, Centella asiatica, also used for the medicinal purposes in many countries around the world. Oh, now this is a brief outline of my presentation, not to so that you know what I'm talking along my slide presentation. I will talk about a little bit of background, introduction, methodology, result discussion, and course conclusions of my work. And this work is actually with my former student around 10 years study, and I have done a lot of work actually from ecotoxicological point of view, and this is to justify and to provide evidence and results. Now, whether these enzymatic activities are good biomarker, okay, of metal pollutions in the plant. So just a brief introduction about what we are doing in our laboratory. So in times of metal stress, so the cells will be under a conditions that need to counteract the stress. So the plant will have a raised level of ROS most of the time. This is known as reactive oxygen species, and this ROS is actually the biomolecules that are actually harmful to the cells that will cause cell injury. These biomolecules will include the hydrogen peroxide, which is harmful to the cell. So it need to be discarded or regulated. And besides hydrogen peroxide, there are also singlet oxygen and what we call as these hydroxyl radicals. So this plant actually developed mechanisms of tolerance to regulate those harmful effects due to the existence of this ROS that is manifest by what I'm saying as a hydrogen peroxide. So basically, there are two mechanisms. There are two the ways mechanism to regulate or to reduce the harmful effects the primary will be the enzymatic antioxidants, now such as catalase CAT, superoxide dismutase, SOD, APX, and GPX. Now alongside with also others we known as the enzyme-like, okay, and the oxidants as well. Now the secondary mechanisms are known as a non-enzymatic antioxidants, and this is actually the low molecular mass, okay, such as tocopherols, prolines, okay, and also this glutathione S transferase. So both mechanism, enzymatic and non-enzymatic antioxidants, they work together in order to reduce the stress. So before it become, we call toxicity to the cell. So these are a very important basic knowledge. So in our work, we try to know if for selected enzymatic activities, which are CAT, SOD, GPX, and APX, are actually can be used as a biomarker for selected metals such as copper stress and also the lead stress under laboratory conditions. So this slide very clearly shown to us that now under heavy metal stress, so the plant will be manifest in three different perspectives. The first is biochemical effects, as what I detailed just now, followed by these are physiological effects, so such as the dotted morphology of the plants, leaves, corrosis, necrosis. So this going to be another aspects from a toxicological point of view. So we can, for example, disturb membrane. We can look at like a camera to see the histological aspect, and the genotoxic effects at the molecular level, which is also what we call genotoxic ecotoxicology, which is very interesting as well. So my work is focused on the enzymatic and non-enzymatic antioxidants. So overall, in the presence of heavy metal stress, there will be an induction of ROS, reactive oxygen species. So this ROS need to be regulated and need to be controlled. So why? Because ROS will actually make the metals such as transition elements, okay, in a single step reaction generating free radicals. So once these free radicals have been generated, so it will disturb the normal metabolism of the cell in the plant. So they will also alter or change the metabolic pathways, especially the thylakoid membrane. So again, they need to be discarded as soon as possible, okay, by the formations of the antioxidant, whether enzymatic or non-enzymatic. And then the ROS will also potentially make the antioxidant enzyme inactive. So whether the numbers are higher or the levels are lower, so this will determine whether they win the match, okay, when they try to drive this ROS away from the living cell. And also heavy metals tablets, okay, will also disturb the antioxidant like glutathione that will challenge the heavy metals. So this slide to show you the basic, again, understanding of when a toxic metals entering the cell through the transporter on the cell wall. Now, as we know that two major characteristics that differentiate the plant cell from the plant cell from the animal cell are the existence of cell wall and the vacuoles. So through the cell wall, when the toxic metals has been successfully transported inside the cell of the plant, so it will be bind to phytogelatins, which is also have been known to be a biomarker, okay, of metal pollution, metal detoxification system in the cell. So if we look at, you know, ROS, okay, in the center here, you may see that ROS will be raised up levels, and ultimately the biomolecules, hydrogen peroxide H2O2 will be given out, and this H2O2 hydrogen peroxide need to be controlled and regulated. And most of the time, okay, the first defense mechanism, okay, by the plant is the antioxidant enzymes. So therefore, based on this basic understanding, we want to know if our work is actually in tally with the theory and based on the copper stress and the PB stress on 4 focus antioxidant in our work, which are the SOD, CAT, GPX, and APX. So toxic heavy metals in medicinal plant is actually a global concern because many countries actually use the herbal plants, and Centella asiatica is one of them. And without much public, information whether these herbal plants is actually safe to be consumed. So 80% of the world population are using the medicinal plant. So public concern is highly of the major focus, okay, on the medicinal plant. And in my work for I think more than about 20 years, I'm working on biomonitoring, okay, of toxic chemicals in Malaysia. And then why is like this Centella asiatica? So basically, if it has view or it has a good characteristics to be used as a biomonitor, as being assumptively as a single species. So we use the word biomonitor, such as net accumulator of pollution or pollutant, large enough for meta-analysis, abundant, easy to identify, tolerant to environmental variations, and of course it is sedentary after it grow in a specific area. Okay, so the objective of this overall work is to determine the relationship of metal accumulation and the antioxidative activities of the 4 enzyme, okay, as I said just now, and to determine the tolerance and toxicities of these metals, okay, and the effects on the plant's antioxidative levels, okay, under laboratory condition. So for methodology, we collect the samples actually around 16 sites in Peninsular Malaysia, and later on it will be analyzed for the 3 metals, okay, in the different parts of the plant and also in the soil, and also the plant parts will be analyzed for antioxidant levels in the leaves and roots, focusing on the 4 enzymes as I said just now. So we did toxicity testings under the greenhouse conditions, and then this toxic test will be done in a hydroponic aquatic media conditions. And then similarly, when we analyze for metals using AAS and the antioxidant analysis as well. So we have done this work for 1 week, 2 weeks, 3 weeks, and 6 weeks. And what we can find is that for the first objective, so we find the sample Centella collected from the wild. Okay, we find that it has some interesting significant correlation. For example, copper with the GPS, copper with APX, zinc with SOD, PB significantly correlated positively with GPX and APX. So this to shows that the raised level of copper in the tissue, okay, in these leaves of the Centella will followed by higher levels of GPX and APX, and also high levels of zinc in the leaves, followed by high levels of SOD. Similarly, high levels of PB in the leaves of Centella will be followed by significant high levels of GPX and APX. So we need a verification whether this also can be found, okay, in our laboratory condition, okay, because in the field there are so many factors can affect our results, ranging from abiotic factors such as the moisture, the climate change, okay, the temperature, to biotic factors such as mutations and also other diseases that is very complicated, okay, to verify, you know, if we just collect from the field. Therefore, we try to do it similarly, okay, under laboratory conditions. And this is the results from the field collected sample based on the roots part. So interestingly, the roots part all show significant positive correlations with the three methods together, the increased level of the four enzymes investigated in this study. So what we can understand from our work is that ROS was produced when exposed to stresses, and this claim can only be verified when we have done the work based on laboratory hydroponic conditions. Okay, this would rapidly attack all types of biomolecules. So therefore, increased activity of the antioxidative system was required, so to protect the plant from the harmful ROS. So as what I explained in a previous slide. Now, the synchronized actions of SOD, CAT, GPX, APX were activated to work with against ROS. So all these different mechanisms are activated in order to reduce the harmful biomolecule effects, especially the hydrogen peroxide of the ROS. Okay. So the second objective is a laboratory condition study. So we found positive correlations, okay, between the leaves and stem to certain pairwises, for example, copper with copper level in the leaf stems, copper and zinc levels in the leaf stem, and almost 100%. Okay, actually it's not 1.00, it should be 0.99. Okay, so it's very correlated, highly correlated. So, and then interestingly, when we try to compare, okay, what we found in the laboratory conditions compared to the field conditions. So this is what we found. The laboratory experiment shows that, you know, okay, the zinc is actually highly correlated significantly in a positive manner. Okay, zinc with SOD, CAT, GPS, APX, and compared with the sample collected from the field. So we see that, okay, zinc only positive correlated, okay, with the SOD. Okay, so there is a discrepancy between the laboratory experimental results and the field collected results. I think this is I think not to say well predicted or expected, but because of so many complicated biotics in the field collected samples. So however, in our laboratory results, okay, experimental results, it shows that, you know, this is what we found when everything are well controlled. So only the metal, the zinc, for example, the zinc, copper, and lead, okay, given higher levels, okay, in the solution of the hydroponic media. So, however, when you look at, you know, out of 12 pairwises, okay, in this laboratory experimental results, there is 6, okay, pairwises that shows positive correlation, okay, of metals, whether zinc or Pb, with the 4 different enzymes here in the leaves, and also about 4 pairwises for the root antioxidants, okay, with the 4 with at least 3 enzymes. This at least indicates that, you know, they can be used as biomarker, okay, for the metal stress. And of course, further study are still much needed. So, however, we need the funding, okay, to do the work continually. So it is difficult for us to get continued funding for now, actually. Okay, go to next. Okay, now in roots, zinc was actually shows significant correlation with GPX and APX. High level concentrations of zinc in the root may reduce uptake of other essential So there is known to have we call antagonistic effects as well. When one metal is higher, the other metals will be lower in the uptake or accumulation of metals. So it is very interesting. So the result could be synergistically or additive. So this actually was supported by some other study where it was also found that the presence of one metal influences the uptake of another. So copper shows significant correlations with the SOD, GPX, and this shows that copper accumulation will triggers antioxidant enzyme. So this is verified as what we see in the previous slide that whether it's true or not based on Centella asiatica. So the results show positively. So will trigger antioxidant enzyme activity in roots due to sensitivity of plant towards the copper toxicity. And this was supported by the result of other study. So what I can conclude, higher accumulation copper lead, especially observed in the roots of the Centella asiatica than the leaves. So the roots is the first organ, okay, to accumulate the metals, okay, compared to the stems and the leaves because it's a close contact to the soil as well as the environmental media. So it is justified and well supported by many studies in the literature. Synergistic effects of copper and lead stress were found at higher metal level exposure. And the copper and Pb stress triggered the increment of 4 enzymes investigated focused in this study in both parts, leaves, and the roots of Centella asiatica. Now, the higher levels of copper and lead, especially in the roots of the Centella asiatica, actually reflected contamination of hydroponic media by these two metals. Of course, bear in mind that if the metals are too high in the media, so I think there is no way any defense mechanism, okay, can be can defend, okay, the cells from injury. So this is at the stress level where everything still can be controlled by the higher levels of the antioxidants. So the increment of the 4 these enzyme activities indicated that Centella asiatica responded to the metal stress by producing, okay, this antioxidant. Now, these activities are significant antioxidant defensive mechanism or system to the copper and zinc, at least based on our study. So it making higher tolerance to toxicity, which are caused by at least these 2 metals, okay, stresses. So collectively, these 4 metals are good biomarker, okay, of a copper and lead stress in the roots and the leaves of the Centella asiatica. In our work, has been recently, I think not recently, last year, published in this stresses under MTPI publisher. So with that, I thank you very much for your attention. And you have any comment, inquiries, most welcome. So thank you.
Thank you, Doctor, for your valuable presentation and presence. Any questions? So I hope no questions. Once again, thank you for your valuable presentation.
Thank you, thank you, thank you.
Moving on to your next session by Dr. Bayu Asma Hello from Bolo University, Ethiopia, in the talk of mapping and determinants of inadequate minimum dietary diversity among children aged 6 to 23 months in 33 sub-Saharan African countries. Hello, Dr. Bayu. Are you ready with your presentation?
Yes, yes.
Okay, now I'm going to make you the presenter.
Okay,
Please click on the screen button to share your screen.
okay.
Yes, we can able to see your screen. Please make it as a full screen.
Can you see my screen?
Yes. Please click on the slideshow toolbar. In the top of the toolbar, there is a slideshow button. Please click on that. On the top of the toolbar,
Cannot maximize my slide.
please click on the slideshow button. It is on the top of the toolbar. Did you see the file, home, insert, design, transition, animation, and slideshow? Yes.
Can you see?
Yes, we can see your screen. Okay, it's okay. Please continue with your talk. Yes, perfect.
Okay. Thank you. Can I start?
Yeah, you can start.
Okay, thank you. My name is Bayuas Mamo. I am from Ethiopia. I am working at Follow University. Today I will discuss in my research entitled Mapping and Determinant of Inadequate Minimum Dietary Diversity among Children aged 6 to 27 in Sub-Saharan Africa. I will start in my introduction parts. Inadequate feeding practices are primary cause of child malnutrition. Inadequate feeding practice is a significant challenge on poverty reduction, adequate dietary diversity, long-term socioeconomic development. Early childhood malnutrition is linked to poor diet quality. Globally, about 88% of countries suffer from 2 or 3 types of malnutrition due to lack of adequate nutrition. Due to inadequate nutrition globally, 2 billion people are deficient in micronutrients, 150 million children under the age of 5 are stunted, and more than millions have impaired cognitive development. Recently, poor nutrition accounts for 45% of children mortality, of which Sub-Saharan Africa shares in the highest burden of this mortality, which is included in my study area. Due to the challenge of achieving sustainable development goal by 2030, which is end all types of malnutrition, becoming increasingly important for geographical location and factors for policy prioritizations. So this study is to investigate geographical variations, high risk of factors, consequences, and the maximum potential of changes. I will discuss next in the method parts. In this for this study, I used DHS or demography health data as a data sources. In this study, about 57,219 of children included in this study from 33 Sub-Saharan Africa countries. The list of 8 food groups collected by the DHS and considered in this analysis are breast milks, grain roots and tubers, legumes and nuts, dairy products, vitamin-rich fruits and vegetables, other fruits and vegetables, fresh foods, and eggs. For this analysis, the dependent variables is children who consume at least 5 of 8 food groups are deemed to have meet the minimum dietary diversity or adequately, or have a good feeding practices. Out of 8 food groups, who consumes 5 of them, at least 5 or 6, 7, or and above are which the children get adequate minimum dietary diversity or good feeding practices. below 5 feed groups is bad feeding practices. The outcome variable labeled as either 0, meeting adequate minimum dietary diversity, or 1, not meeting adequately minimum dietary diversity. For this analysis, use two different models. The first one, geospatial models, and the next is regression model or factor analysis models. In the geospatial models, use three sub-models. The first one, hotspot analysis. The hotspot analysis used to identify statistical significant spatial cluster of high value hotspot and cold spot. For this analysis, use GeodaGIS. The GeodaGIS used this analysis. The hotspot and cold spot identifies the polygon-based or region or province-based analysis, the prevalence of region or province compared to their surrounding neighbors of regions. It categorizes at either hotspot or cold spots. The next is spatial scan status analysis. The spatial scan status analysis higher aggregate concentration of instance were included in the circular windows. For this analysis, use StatScan and QGIS, both QGIS and StatScan. For this analysis, based on circular enumeration areas, based on the number of cases and the number of samples in the enumeration area, the higher burden or the higher concentration of cases found in the circular windows. The next is the last is spatial interpolation. The spatial interpolation based on measured area predicts the proportion of unmeasured area. For this analysis, used SagaGIS. For this analysis, used to predict unmeasured area based on the nearest neighbors of measured area or enumeration area prevalences of inadequate minimum dietary diversity. The next is for factor analysis. The factor or regression analysis. In the level analysis, for the DHS data is used for level analysis. Individually, it needs to do within household. Household needs to do community. Community is needs to do region. Region or province needs to do country. So, such type of data used to two or more level of analysis. But in this analysis used only two level analysis. The lower level analysis, maternal or household, and the higher level is community or enumeration area. For factor analysis, binary logistic regression used. The child who have not give 5 or more group of food in the preceding of 24 hours. For the consequence, model, anemia, stunting, and wasting are the consequence of inadequate minimum dietary diversity. For such analysis, anemia, stunting, and wasting individually as a dependent variable, and minimum dietary diversity is one of independent variables with adjust with other variables like socioeconomic factors and others. Each case, separately, the dependent variable and the minimum dietary is one of the independent variables. Before beginning any investigation, all data from all countries are combined into a single dataset, not each separately analysis. It is all in one. For all findings, weighted is implemented because DHS data have each individual or observations have its own weighted variables. So based on the weighted variables, weighted method is implemented. In this next, I will discuss in the result parts. For the first result indicates that the bar chart indicates the first green color indicates a child who consumes 5 or more food groups or good feeding practice found in these proportions. For the in average in sub-Saharan Africa, about 20% of children who have consumed adequate minimum dietary diversity. And the next green is three food consumes 3 food groups, and the next orange color indicates that 2 food groups, and the yellow color indicates that only 1 food group was consumed, and so on. In this analysis, this bar graph with confidence interval indicates that in the residence and in the age groups. The first indicates that the red color indicates the rural residences who consume or who have good food practice, and the blue color indicates that the urban residences. In the urban residences, the maximum found is in Kenya. 47% of children, over about half of children who live in Kenya, have found this in adequate minimum dietary diversities. And the next is Malawi and Rwanda. Children from Malawi and Rwanda, out of 5 children, about two of them were found this adequate minimum dietary or have good feeding practices. And the lowest indicates that in Burkina Faso, Liberia, and Ivory Coast, which accounts about out of 10 children, only one of them were found this adequate minimum dietary diversity. In rural regions, the maximum indicates that in Kenya, South Africa, and Mozambique. In Kenya, South Africa, and Mozambique, all about children from those areas, 28% of them were adequately minimum dietary diversity. According to age, below 1 year and above 1 year. Below 1 year is above 1 year where have good feeding practice compared to below 1 year. For this map indicates that the first two maps indicate based on polygons. The first one is pure prevalence, and the second is hotspot analysis, or hotspot and cold spot analysis. In the first analysis, the bold green, children who live in this bold green regions were found. Out of 10 children, only one of them were obtained adequate minimum dietary. The rest is not get adequate minimum dietary diversity. And the next red color indicates 18% to 90% of children who live in this color found this adequate minimum dietary diversities. The next polygon based analysis or hotspot analysis used to GeoData, the red color indicates that the hotspot. That means a child lives in this red color indicates that the highest burden of inadequate minimum dietary diversity compared to their surrounded, and the blue color indicates is the lowest inadequate minimum dietary diversity compared to their surroundings. The next two maps indicated based on enumeration area or point data prevalence. The first one in the circular windows indicates used to SAT-scan. In the circular windows used to the concentration of cases or inadequate minimum dietary diversity in the circular windows, the higher concentration found in the circular windows. The first one is a child who live in this area, 85% of them suffers inadequate minimum dietary diversity. Those relative risk is 1.3, and the second found is at here. In this circular windows, 82% of children suffers in adequate minimum dietary diversity. The last map indicates that interpolation. The interpolation used to predict and measured area based on the nearest neighbor of measured area. For this analysis, based on enumeration area prevalence, about the bold green colors indicators approaches to all students suffers inadequate minimum dietary diversity, and the bold green indicates that all children found adequate minimum dietary diversity. For this indicates that multi-level logistic regression. In multi-level logistic regression indicates that the vertical line indicates in the one from this vertical line, the left to from this vertical line, the reference is risk, and the right is the reference is protective. For example, in the first, mothers whose work status is mothers have no work is adjusted loss ratio is 0.81. This indicates that children from mothers who have no work is from about 19% more likely to suffer or from inadequate minimum dietary diversity compared to child comes from whose mother have work, and so on. This is the consequence model. In the consequence model indicates that due to inadequate minimum dietary diversity, the child will anemic, wasting, and stunted. For this indicates that the first one is a child who have not get adequate minimum dietary diversity 2 times high risk for anemia who have not get adequate minimum dietary diversity and wasting and stand like that also. In this, the first bar chart indicates that inadequate minimum dietary diversity proportion of inadequate minimum dietary diversity or in each food groups. In Sub-Saharan Africa, 86% of children who have not consumed or who have not get eggs, and 18% of children who have not consumed or get other fruit and vegetables, and so on. The nice finds of gates of children in Sub-Saharan Africa is breast milks, which account 80% of children in Sub-Saharan Africa gets breast milks. The next bar graph indicates that the percentage of the change of percentage of any concerned body or governments working in hardly for child feeding practices is the maximum potential of increasing indicates that by this bar charts. The red color indicates the horizontal red color indicates now the average prevalence of child's adequate minimum dietary diversity in Sub-Saharan Africa, which account 20%. When we work in legumes and lettuce, can be reach 34%. The maximum in single food practice items, we work in single food practice or items, the maximum contribution found is in 37%, can be changed 70% when works in eggs. And the other two food groups works in eggs with other fruits, vegetables available in Sub-Saharan Africa for each children can be reaches the prevalence, the percent of adequate minimum dietary diversity from 20% to 61%, increased by 41%. When 3 food groups works, the maximum is legumes and lettuce, eggs, other fruits and vegetables, reaches 84% of Sub-Saharan Africa children's adequate minimum dietary diversity, which changes 64% increased. It is all about my findings. Next, in the discussion parts. Inadequate minimum is serious public health problem for all countries. So in Sub-Saharan Africa, spatial heterogeneity occurs both between and within countries. Inadequate minimum is the tangible cause of child anemia, stunting, and wasting. More efforts on maternal changes for advantageous characteristics can be improved easily. Thank you. It is all about my research. Anyone who have a question, I can accept it.
Thank you, Doctor, for your presence and presentation. Is anyone having any questions?
Hello.
Hello.
Yeah.
Yes, Doctor.
Yeah. Hello. Hello. So I want to ask a question. There is an imperative way to recognize the malnutrition effect on the child and other people's.
What?
Actually, if you want to prevent this, so there is some strategies, easy strategies to control the malnutrition among children's.
It is not clear to me your question.
Hello.
Hello, can you hear me?
Yeah. Hello. You hear me?
Yes.
There is some imperative strategies to control the malnutrition in children's.
Okay. It is working in minimum dietary diversity. Malnutrition is the cause of this inadequate dietary diversity. So, in this research, I shows the inadequate minimum dietary diversity and the consequences or the outcome of inadequate minimum dietary diversity. Malnutrition is the cause of it, which is anemia, stunting, and wasting is the outcome of malnutrition or inadequate, the result of inadequate minimum dietary diversity.
Okay.
Thank
Yeah.
you.
Thank you.
Thank you.
Thank you, Dr. Bayo, for your presence and presentation.
Thank you.
Thank you. So let's move on to our next session by Dr. Amit Kumar Keshavane from Indian Agricultural Research Institute from India. The title of Visualizing the Infection Process of Xanthonas compestris PV campestris in susceptible and resistant host of Brucea species causing black rot diseases. Hello, Dr. Amit Kumar. Are you ready with your presentation?
Yes, ma'am. Hello. I'm ready.
Yes. Okay, so now I'm going to make you the presenter. Please
Yeah.
click on the screen button to share your screen.
Okay.
Yes,
Yeah, ma'am.
we
Now
can
it
able
is
to.
visible.
Yes, it is visible to us. Yes, perfect. Please continue with your talk.
Yeah, ma'am. First of all, a very good morning to all of you. Myself, Amit Kesarwani, and doing PhD in biotechnology with the joint collaboration of Indian Agricultural Research Institute, USA, and Amity University, India. So today I am here to present my research work on visualizing the infection process of Xanthomonas campestris, Pathover campestris, in susceptible and resistant host of Brassica species causing black rot disease. So this is a part of my PhD research work. My research motivation indicating Xanthomonas campestris, Pathover campestris, is a major bacterial pathogen that infects most of the cruciferous crops, including cauliflower, cabbage, and other vegetable crops. And the disease causes substantial damages up to 10 to 15% on yearly basis. The genus Xanthomonas, including most economically important pathogenic bacteria that are generally associated with vegetable plants. And the taxonomy of the genus was initially determined according to the host preference, and consequently large number of species and pathogens have been defined. Now present. So presently 11 races of Xanthomonas campestris, Pathover campestris have been identified with the host specific interaction that causing black rot disease in crucifer crops. So the pathogen generally Xanthomonas campestris, Pathover campestris is a gram-negative rod-shaped bacterium and the causal agent of black rot disease in crucifer crops, and it is a systemic vascular pathogen that producing V-shaped yellow lesions starting from the leaf margins and blackening of the veins. In XCC, many genes or proteins are potentially involved in the virulence or pathogenicity development in crucifer crops, and the genome annotation of this pathogen possesses at least 9 AVR genes, that is AVRBS1, AVRBS2, ZOP-E, ZOP-H, ZOP-AC, ZOP-AG, ZOP-AH, AVR-XCCA, and AVR-XCCA2. And in my PhD research work, I have characterized the functional role of AVR-BS1. So the second picture demonstrating the V-shaped lesion of XCC that is represented by star marks. The objectives of this study represent the development of green fluorescent protein expressing Xanthomonas campestris, Pathover campestris, inoculation of XCC, P. cambia, MGFP in susceptible Brassica oleracea variety Pusa Sarah, and resistant host Brassica juncea variety Pusa bold of Brassica species. And the final objective was to demonstrate the visualization of the colonization of Xanthomonas campestris, Pathover campestris by confocal microscopic analysis. So the methodology initiated with the inoculation of a single colony of Xanthomonas campestris pathover campestris in 50 ml urea broth and grow at 28 degrees centigrade for 24 to 48 hours to obtain an optimum OD, and then prepare the electrocompetent cells. So the method of this study uh, has been divided into two steps. So the one step is step one is representing the preparation of electrocompetent cells of Xanthomonas campestris by Wong et al. Method. And in this step, we have transformed the Pcambia 1302 harboring MGFP gene in XCC by electroporation, and then selected the positive clone of GFP expressing XCC cells. In the second step, we have inoculated the Xanthomonas campestris, pathover campestris, cause expressing GFP protein in susceptible and resistant host of Brassica species. Then, after different time intervals, we have visualized the infection process of XAC through confocal microscopic analysis. So, overall, the preparation of GFP expressing cells, yeah, GFP expressing bacteria by using 1 microgram of plasmid of Pcambia 1302 and electroporate it in Xanthomonas campestris pathover campestris electrocompetent cells via electroporation method, and the recombinant cells were selected on Luria agar supplemented with 50 microgram per ml kanamycin. These clones were verified by PCR detection of MGFP gene through conventional PCR method, and these positive clones were used for the analysis of GFP expression in host and resistant and susceptible host. So now we are inoculating the Xanthomonas campestris pathover campestris by syringe infiltration assay in susceptible and resistant hosts of Brassica species, and observing the disease or development in these hosts. So after different time intervals, we have collected the samples of Brassica juncea and Brassica oleracea, that is 24 HPI, 48 HPI and 0 HPI. So finally, we have visualized uh, the bacterial cells in the different parts of the plant by confocal microscopic analysis. So in control, we have clearly seen there is no any cells of the bacteria present, that is treated with the saline buffer. And in our experimental host, that is a susceptible host of Brassica oleracea variety Tussa we have clearly seen we are clearly seeing the bacterial cells in seed, root, stem and leaf, and with the time the number of cells and the replication of bacteria was increasing. So we are going to the next part of our study. So now here we present the migration of the Xanthomonas campestris pathover campestris in Brassica juncea, that is a resistant host of Brassica. So in control there is no cells, but in experimental tissue we have analyzed very less amount of bacterial cells represent in the seed, root, stem and leaf, at least after 48-hour post inoculation. So this study reveals that the analysis of GFP extract bacterial cells, the pathogen was migrated to leaf, stem, root, seed through vascular tissue in susceptible host, while in resistant host, the movement of the bacteria was restricted. So these results are also represent the XCC is seed-borne pathogen, and the method will also be used for the localization, the pathogenicity or AVR proteins of bacteria in host by GFP fusion with targeted proteins. These are the references, and we have recently published the whole genome sequence of Xanthomonas campestris pathover campestris race 4 isolate causing black rot disease in Brassica oleracea variety capitata in Plant Disease Journal, and also published in NCBI database. And last not least, I am thankful to Science and Engineering Research Board, Department of Science and Technology, Government of India for providing me the financial support to achieve one of my PhD research work under the project entitled Identification of Resistance Source Against Prominent Races and Functional Characterization of A-virulent genes of Xanthomonas campestris, Pathover campestris, causing black rot disease of crucifer crops. And I am also delighted to thanks to Coalesce Research Group for providing the opportunity to present my research work in the International Conference Plant Biology. Thanks to all and namaste.
Thank you, Dr. Amit Kumar, for your presentation. Is anyone having any questions?
Yes, Dr. Kumar, can I ask you a few questions?
Yes, sir.
Okay, you are working on Brassica juncea. So what about other species like Brassica raba that are also very popular species in tropical area, including in
Yes, sir.
Malaysia?
Yes, sir. It is a susceptible host of Xanthomonas campestris.
Okay.
Actually, yes, sir.
The, yeah.
There are different genus: Brassica napus, Brassica rapa, Brassica oleracea, Brassica juncea. So we have screened in different genus and found that Brassica juncea is a resistant host against Xanthomonas campestris, Pathover campestris.
Okay, now in order to tackle the problem of diseases, infections on these popular vegetables in tropical countries, so
Yes, sir.
having your study results, so in order to increase the crop plantation, in order to cater the food for the poverty people, so what about the use of chemical fertilizer? So what do you think?
Sir, I don't understand your question. Chemicals?
Okay, now the infection diseases on the vegetables like Brassica species
Yes, sir.
is a problem. Okay, maybe your work can reduce the problem. So
Yes, sir.
what about
No,
the
sir.
use
No,
of,
no. Sir, my work
yeah?
not reducing. Now I am presenting the localization of bacteria in susceptible and resistant hosts of Brassica. It is generally the way to identify the bacterial cells in host through the confocal microscopic analysis.
So are, are your research output going to reduce the problem of the black rot disease?
Yes, sir. So to reduce the problem, we have screened the source of resistance against XCC. And recently we have identified 7 out of them in Brassica oleracea groups.
So I
That
believe
is
your work is,
including early and mid crops.
yeah, so I believe your work, your result output going to increase the yield and the productivity of Brassica by reducing the disease of the Brassica. Am I correct?
Yes, sir.
So all the best to your work. Okay, so thank you.
Yeah, welcome, sir.
Thank you, Dr. Amit Kumar, for your information. Is anyone
Thank you,
having any
ma'am.
questions? So I hope no questions. Once again, thank you for the valuable presentation.
Thank you. Thank you. Thank you to all of you.
So let's move on to our next session by Dr. Shama Rahmi Divin from Siraj University from Iran in the title of Bioremediation Technology and Agriculture. Hello, Doctor. Are you ready with your presentation?
Hello, hi.
Yes,
Yes,
hi. Are you ready with your presentation?
yes.
So now I'm going to make you the presenter. Please click on the screen button to share your screen. Okay, we can able to see your screen.
Hello, everybody. Um, In the name of God, my subject is about bioremediation, technology, and agriculture.
Please click on the right arrow to change your slide.
Soil pollution is a worldwide problem that draws its origin from anthropologic and natural sources. Urbanization, industrialization, and food demand increases have required the use of compound substances and chemical agents which over the years have brought on the dispersion and accumulation of pollutants in the environment. The common pollutants present in the soil are heavy metals, polyalkylic aromatic hydrocarbons, or pesticides.
Hello, Dr. Samar Rahmi. Can you hear me?
Yes, sorry, my computer is hang up.
Okay.
I prepare in another computer.
Okay. So due to some technical issue, let's wait for a few more minutes to continue the talk. Hello, doctor. Hello, Dr. Shamarami. Can you hear me?
Hi. Yes. Sorry. I continue.
Okay. Now I'm gonna make you the presenter.
Yes.
Please click on the screen button to share your screen.
Yes.
Yes, okay. Please continue with your talk.
Sorry. Soil pollution is a worldwide problem that draws its origin from anthropologic and natural sources. Urbanization, industrialization, and food demand increases have required the use of compounds, substances, and chemical agents, which over the years have brought on the dispersion and accumulation of pollutants in the environment. The common pollutants present in the soil are heavy metals, polycyclic aromatic hydrocarbons, or pesticides. After 1945, there was a rapid development of the agrochemical field characterized by the introduction of many insecticides, fungicides, herbicides, and other chemicals to control pests and ensure the yields of agricultural production. They are also used to control vector-borne diseases such as malaria and dengue. Moreover, pesticides are applied in aquaculture, horticulture, and for various general household applications. From 1990 to 2018, There have been registered amount of used pesticides by all countries in the world, especially in Asia and America. Looking at the types, fungicides and bactericides are used more than the others. For these reasons, the residual pesticides concentration present in the soil must be reduced after effective remediation techniques must be used to do this. An eco-friendly, cost-effective, rather efficient method is bioremediation, which is an alternative to more expensive and toxic approaches such as chemical and physical methods. In bioremediation, the removal can be achieved by exploring the microbial activity of microorganisms. The microorganisms, primarily bacteria or fungi, transform pesticides into less complex compounds, CO2, water, oxides, or mineral salts, which can be used as carbon, mineral, and energy source. Bioremediation relies on stimulating the growth of certain microbes that use contaminants like oil, solvents, and pesticides as a source of food and energy. Effective bioremediation needs a combination of the right temperature, nutrients, and food. Otherwise, it may take much longer for the cleanup of contaminants. If conditions cannot favorable for bioremediation, they can be improved by adding amendments to the environment, such as molasses, vegetable oil, or simply air. These amendments create optimum conditions for microbes to flourish and complete the bioremediation process. Biological remediation can be categorized into two types: microbial remediation and phytoremediation. Microbial remediation. Microorganisms are well known for their ability to break down a huge range of organic compounds and absorb inorganic substances. Currently, microbes are used to clean up pollution treatment in processes known as bioremediation. Microorganisms are readily available, readily characterized, highly diverse, omnipresent, and can use many noxious elements as their nutrient source. Although many microorganisms are capable for degrading crude oil present in the soil, it has been found beneficial to employ a mixed culture approach than the pure culture in bioremediation, as it shows the synergetic interaction. The bacteria that can degrade major pollution include the Pseudomonas, etc. The phytoremediation is a bioremediation process that uses various types of plants to remove, transfer, stabilize, and/or destroy contaminants in the soil and groundwater. There are several different types of phytoremediation mechanisms: rhizosphere biodegradation, phytostabilization, phytoaccumulation, rhizofiltration, phytovolatilization, phytodegradation. Rhizosphere biodegradation. In this process, the plant realizes natural substances through its roots, supplying nutrients to microorganisms in the soil. The microorganisms enhance biological degradation. Phytostabilization. In this process, chemical compounds produced by the plant immobilize contaminants rather than degrade them. Phytoaccumulation, also called phytoextraction. In this process, plant roots absorb the contaminants along with other nutrients and water. The contaminants mass is not destroyed but ends up in the plant shoots and leaves. Rhizofiltration is similar to phytoaccumulation, but the plants used for killing up are raised in greenhouse with their roots in water. Groundwater is pumped to the surface to irrigate these plants. As the roots become saturated with contaminants, they are harvested and disposed of. Phytovolatilization. In this process, plants take up water containing organic contaminants and release the contaminants into the air through their leaves. Phytodegradation. In this process, plants actually metabolize and destroy contaminants within plant tissues. Best plants for phytoremediation: Indian mustard, willow, poplar tree, Indian grass, sunflower. Some examples of bioremediation-related technologies: permeable reactive barrier, biosparging, bioventing, bioaugmentation, biostimulation, biopiling, bioreactor. Permeable reactive barrier are an in situ technology applied to remediate groundwater contaminated by many types of pollutant. A permanent or semi-permanent reactive barrier composed mainly of iron is immersed in the contaminated groundwater stream. Biosparging involves the injection of air under pressure below the water table to increase groundwater oxygen concentrations and enhance the rate of biological degradation of contaminated by naturally occurring bacteria. Bioventing involves controlled stimulation of the airflow, providing oxygen to increase microbial activity and consequently enhance bioremediation. Bioaugmentation is the introduction of a group of national microbial strains or a genetically engineered variant to treat contaminated soil or water. This method can be used because the native microbial population may not be able to destroy the damage caused by complex substances such as oil. In biostimulation, native microorganisms are stimulated to grow with the addition of growth factors such as nutrients like phosphorus and nitrogen. Biopiling is an ex situ bioremediation technology that has been extensively used for remediating a wide range of petrochemical contaminants in soil. Biopiling involves the assembling of contaminated soils in the piles and stimulating the biodegrading activity of microbial population by creating near optimum growth conditions. A bioreactor can be defined as a system in which the conditions are closely controlled with respect to physiological conditions to induce certain behavior in living cell, tissue, or organ. A bioreactor may be used as a device to create conditions for the removal of water and soil contaminants using microorganisms. Limitation of bioremediation. Not all compounds are susceptible to rapid or complete degradation. There are some concerns that the product of biodegradation may be more present or toxic than the parent compound. Bioremediation often takes longer than other traditional options such as excavation and removal of soil or incineration. Bioremediation is used for the remediation of metal, radionuclide, pesticides, explosive fuel, volatile organic compound, and semi-volatile organic compound, if may be used to clean up contamination found in soil and groundwater. For radioactive substance and chelating agent are sometimes used to make the contaminants amenable to plant uptake. Thank you for your attention.
Thank you, Dr. Rahmi, for your presence and presentation. Is anyone having any questions? So I hope no questions. Once again, thank you for your presentation.
Thank you.
So let's move on to our next session by Dr. Daud Delegates from Flexible Hopto Biotechnology University from Cote d'Ivoire, in the title of Bioactive Peptides from Sprouter Vandos with Anti-Diabetic Potential by Inhibition of Alpha-AMC and Alpha-glucoside Enzymes. Hello, Dr. Gladys, are you ready with your presentation?
Hello.
Hello.
Hello. Do you understand
Hello.
me?
Yes, we can
Yes.
hear you. Are
Yes,
you ready
I'm
with your
ready.
presentation?
I'm ready for my presentation.
Okay, so now I'm going to make you the presenter. Please click on the screen button to share your screen.
Okay. Comment est-ce qu'on fait? How can screen?
Did you get the pop-up window?
I don't know how to
Just click
share
on the
my let
screen sharing button.
share. Okay.
Yes, we can able to see your screen.
Okay. Thank
Okay,
you.
perfect. Yes, please continue with your talk.
I can present.
Yes, please start.
Okay. Good morning, ladies and gentlemen. I am Dr. Doué Ginette Gladys from Félix Houphouët-Boigny University in Côte d'Ivoire. The topic, please. The topic I am about to address is bioactive peptides from sprouted vanzou, a neglected local legume with anti-diabetic potential by inhibition of alpha amylase and alpha glucosidase enzymes. To introduce, we can say that diabetes is a worldwide public health problem, and Africa is not accepted to this trend because this disease is rising sharply in most of African countries. Therefore, there is a need to reduce this disease rate in order to meet the development challenge in Africa. We can note that two critical points have been identified in the development of this disease. They are chronic hyperglycemia and reduction of the body's antioxidant mechanism. So the treatment of this disease should take into account the mechanism involved in the regulation of hyperglycemia through absorption, through inhibition of alpha amylase and alpha glucosidase, two enzymes involved in the blood sugar regulations. We can note today that the drugs involved in the current treatment of diabetes are not accessible to most of African patients due to their low income and to the fact that these drugs have a lot of side effects. This leads researchers to explore various approaches, including functional aspects of food by the bioactive molecules. There's multitude of substances, including peptides, has a significant impact on human health. So for peptides, for having peptide, we need to hydrolyze food protein to obtain these bioactive peptide. We can note that legumes are considered as excellent source of peptides due to their high protein content. Among them, Vuanzu, a local and neglected legume with high protein content, can be exploited for molecular bioactive research. However, the potential of this legume plant, to generate bioactive peptide has been little explored today. So the objective of this study was to enhance the value of sprouted Vuanzu seeds by highlighting the anti-diabetic potential. Concerning the material used in this work, figure 1 show the material used, which is made of Vuanzu seeds, which were brought from a local market in Cote d'Ivoire, and as shown in photography 1. After that, after brought seed, the seed were allowed to germinate at day 2, day 4, and day 6. After the different germination time, the seed were transformed into flowers, and the flowers were delipidated. And after delipidated this flower, we proceed to extraction of protein isolate by alkaline extraction at pH 8.0, followed by isoelectric precipitation at pH 4.3. After protein extraction, we proceed for total protein content evaluation, for simulation of gastrointestinal digestion using successively pepsin and pancreatin enzyme hydrolysis, and then these isolates were fractionated by dialysis through cellophane membrane with 10 kilodalton cutoff. After this, we obtain results that we will show you. Table 1 shows the extraction yield and protein content of the different crude protein isolates. We can notice that for extraction yield, all the extracts, all the isolates displayed yield value above 60%, above 60%. This high yield reflects the efficiency of the extraction method that we used. Concerning the total protein content, We can note that all the crude protein isolate displayed an average of 86%. This reflects to the purity of our extracts, which can be qualified as isolate, as protein isolate. As concerns the potential of this extract on the inhibition of the two enzyme studies, Figure 2 shows the percentage of inhibition of alpha amylase and alpha glucosidase by the crude protein isolate first. We can note that all the crude extracts at D0 of germination and D2 from D6 of germination isolate showed lower inhibitory activity of alpha amylase and alpha glucosidase compared to the control, which is acarbose, a drug currently used in the treatment of diabetes. There's similar results have been reported in the literature for crude extract for soybean and bean on alpha amylase only. When we digested this extract, Figure 3 shows the effect of digested protein extract on alpha amylase and alpha glucosidase activity. We can note that only the crude extract on day 2 of germination show the similar inhibition with the control. The same result have been reported on the literature on protein extract of sprouted soybean at 6 days of germination. When we take these extracts at 2 days of germination and we fractionate it through dialysis and we obtain in figure 4 the percentage of inhibition of alpha amylase and alpha glucosidase of the peptide fraction from day 2 germination. We note that similar results have, we note that only the outer fraction display the same capacity of inhibition of alpha glucosidase comparable to the control acarbose. So we can note that similar results have been reported on the literature and that confirming that there is the low molecular weight peptide that are responsible for this kind of inhibition. To conclude, we can say that germination followed by digested hydrolysis of Wanzhou proteins allows generation of bioactive peptide on alpha glucosidase only. These peptides with small weight molecular cells under 10 kilodalton size and from 2 days germination digested protein isolate show the strong inhibitory activity on glucosidase enzymes. So we can say that the injection of this plant, Wanzhou, can provide bioactive peptide that can manage and reduce the risk of diabetes, and this could be used as functional and nutraceutical ingredient in the treatment of the diabetes. We have finished our proposal. Thank you for your attention.
Thank you, Dr. Coalesce, for your presence and presentation. Any questions? So I hope no questions. Once again, thank you for your valuable presentation.
Okay, thank you.
Thank you. Let's move on to our next session by Dr. Isra M. Abdul Monim from Grand Egyptian Museum from Egypt in the title of Anti-Termitic Activity of 3 Plant Extracts, Chloropetrose and Bioagent Compound against Termitite Microcitomenes Ignotus Silvestri in Egypt. Hello, Dr. Isra. Are you ready with your presentation?
Hello everyone,
Yes, hello, doctor.
I'm
Are you
ready.
ready with your presentation? Okay, now I'm going to make you
Yes,
the
I'm ready.
presenter.
Please share my presentation.
Please click on the screen share button to share your screen.
One second, please.
Okay.
Could you share my presentation again, please?
Just a second. I think she has lost the connection. Let's wait for 2 minutes. She'll reconnect again. Hello, Dr. Isra. Can you hear me? Hello, Dr. Isra. Can you hear me?
Yes, I can hear you very well,
Yes, we can able to see your screen.
but I can't share my presentation.
Okay.
Uh, I can't share my presentation yet.
We can able to see your screen. Please open your presentation.
Where is the icon? I can share it.
Sorry. Please open your PPT file.
PPT file. Oh, okay.
Yes.
You can see it, guys.
Yes. Yes. Please make it as a full screen.
Okay. One second, please.
Slideshow. Please click on the slideshow button.
Okay.
Yes. Please continue with your talk.
Okay. First, I'm so sorry for your waiting.
Okay, it's okay.
My presentation, oral presentation today about study of the effect of termites on the damage of mural painting executed on brick stand building and the methods of resistance applied to one of the tombs of Bir Shaghala New Value, El Wahed, Egypt. My name first is Isra Mahmoud Abdel Menaim. I'm a specialist of restoration in a Grand Egyptian Museum, one of conservator of team of Tutankhamun jewelry, master degree in archaeology restorations, PhD registered in archaeology field 2018, former restoration specialist and archaeological science. The research consists of chapter one. It's 5 chapters, but today I will talk about just three of it. Chapter one: study of mural building in Bir Shaghala. Chapter two: study the effect of termite and fungi infestation on the fibers bearing bricks and photographic layer in tombs Bir Shaghala in the New Valley. Chapter 4, experimental study of the proposed treatment and restoration materials for termites. Chapter 5, applied study in the very Shagala tomb. First, I will talk about this tomb. It's a very unique tomb. It consists of two methods of mural painting. In paint, in color paint. The first method is deep and relief inscription. This method was used only on the threshold of symmetry. The second method, direct drawing method. This method used in the entrance of the cemetery and the hall leading to the burial chambers and two burial chambers. This is the pictures of two methods of painting in this tomb. It's still perceive the colors and it's a huge chambers full of colors. Sorry, 1 second. In these pictures, we can see the chamber, burial chamber, and in these pictures, in front of the tomb. Study of the effect of termite and fungal investigation in the chamber of the Birshagala tombs in the New Value. The termite microterms showed structural damage in Birshagala cemeteries located in the Oasis of Dakhla, Egypt. The mud tubes of this term spread inside and over the mural painting floors of the tomb. Extracted from lavender and incense oils like marjoram and clove were tested for their activity of termites and compared with the bioinsecticide bacillars of chemical pesticides like Rosban and Victoral, and we will see over the slides the comparison between Protecto and Rosban and the oils extracts from plants. This is ants or termites in this tomb, and they made a huge deterioration on the wall of the tombs in burial chambers and inside chambers and outside chambers of tombs. Okay, next slide. A trend towards environmental friendly chemicals for use in termite management has been occurring globally. This study examined 3 natural occurring plant extracts from lavender and marjoram and clove. Against the termite, plant extract results were converted to 2 commercial used termite pesticides, the bioinsecticide bacillars, Protecto 9.4% and Rosban chlorpyrifos 48%. With gas chromatography mass spectrometry analysis was used to identify the main compounds in the 3 plant extracts and the compounds of chemical pesticides. The extracts of oils causes a huge the extract of oil Bulgaria causes the highest termite death rate with almost 770 milligram in liter. Exposure to lavender extract showed a high death rate with 1086 milligram liter. Finally, clove extract didn't show significant activity with an 2000 milligram liters. So we can see here is the lavender and the lavender is showed a high death rate with termites. And is all of this organic plants extracting from organic plants. Besides that, we can see also the effect of drospan and protecto as a chemical pesticides. It's a huge effect of soldiers and workers under the application of these two chemical pesticides. The value of this effect reported for NIFOs were less than 120 and 627 milligram per liter after exposure to drospan and protecto and Vulgare extract. The result of the study shows that some extracts have a low toxicity compared to the drospan and protecto. So we can see the huge effect of drospan and protecto compared to the plants or plants that extracted, but it's useful for we now prefer to use plant extractors to use in these tombs. It's useful for visitors, for archaeologists, and we talk about use it in generally in the next days. Now we can see in these pictures the compound of mid-brick. We can see cellulose materials in servers of Berishagala. First, Berishagala cemetery is located to the west of the city of Mout, 3 kilometers from Dakhla Osis, Egypt. The site huge infestation by the termite microtermites. It's very huge because of building and the soil of tombs. So it's a very huge effect of this insect in the mid-brick of tomb. This termites forage and feed on the cellulose materials used in the constructions of the mid-brick from which the tombs are built. We can see in these pictures, this picture by digital imaging of sample of mid-brick showing the presence of straw in the formation of mid-brick. And this is the second this I will talk about. Show a sample of present of calcite and the sand inside the clay layers. These pictures, these pictures. Sorry. And next pictures is the sample of mid bricks shows the presence of straw These two pictures by digital microscope USB. Terms investigation in Egypt. This terms was reported along the northwestern coast and in the western desert where the Oasis of Dakhla is located. Termite mud tube spread inside and over the mural painted floors of the tombs, causing structural damage that often needs repair. The damage observed in some of monastery buildings. In this pictures by digital microscope photography, we can notice magnifications of 50 tubes escape and feeding on the wall of mid brick inside the tombs in the burial chambers of the cemetery. And these tubes, as we show, these tubes, it's almost 50 tubes. And this is in this tube, we can see this escape and feeding the termite also inside on the burial chambers, as we show. These two pictures also the tubes of termite on the mid brick outside the tomb. We can notice and see that the huge damage caused by termite attack. Picture A, damaged in materials of mid bricks, and we can see the materials is full fill the floors, and the termites tubes between the mid bricks. Picture number B. Picture B, damage to the surface wall and shelter tubes of termites over the mid bricks, as we see. This pictures is very unique pictures. I took it by digital microscope. You can hear me? You can hear me? This is Lina.
We can hear you, doctor. Please continue.
Okay. This picture, as we talk, it's a very unique pictures. I took it by a digital microscope photography with a magnification of magnifications of 200. I use microscopes, the digital microscope inside the tubes. And in this picture, you can see it's found the nymphs and eggs of termites, of termites, and there were also fungal spots inside the tubes, side by side of white ants. In these pictures, in these pictures, we can see the spot of fungi inside the tubes, side by side of ants, and this we can eggs of termites, and this is nymphs of is big. It's big egg of ant, and this is the nymphs of ants of termites inside the tubes. As I show. Next slide, talk about experimental study of the proposed treatment and restoration of materials for termites and fungi investigation. First, I talk about termites, but in this research I talk about termites and fungi. together because I found the terms and fungus side by side in the tubes and in the wall of mural painting and in the my black earth. But now we talk about especially about termites and the situation of it. Treatments of termites used to control the termites include bioagents, chemical pesticides, and natural products. Natural products investigated for termites management include plant extracts. In many parts of the world, chemical pesticides are considered poor for the environment, and the new research is examining the use of natural occurring, less environmentally toxic than plant extracts. In this research, I used 3 plant extracts from spike lavender, marjoram, dried leaves of marjoram, and clove oil were selected for their potential effect on termites. The extractions product was carried out of the Natural Research Center in Egypt. I went to the Natural Research Center in Cairo, and I found these 3 plants extracts and more of this like tea plants and it's huge oils there. Plants was shade air dried in the laboratory or at room temperature for 1 week. Ground into a fine powder in electrical blender. About 100 grams of powder of this powder material was put into 2-liter flask containing 1,000 milliliter of distilled water and extracted by the hydrodistillation method using a lavender tube for 3 hours. This is where I put it. This how I prepared the plants extracted in laboratory in Egypt in Cairo University in Egypt. Collected and reeling the termites, microtermites of termites casts were collected from soil around cemeteries and kept in container with a portion of termites nesting materials. In this tomb, you could find a lot of termites around the tomb. In the soil, so I collected from the soil around the cemeteries and keep it and counter with a portion of termites materials. Briefly, from the experimental location, termites were placed inside a special box for preservation, and the box were covered with wool textile to maintain the temperature, because the temperatures in the OCs are so high, as almost 50 degrees during the transfer to the laboratory in Cairo. We measure the anti-termites activity of the 3 plant extracts, Protect2 and Dorsben. Similar to the laboratory reeling, the materials were prepared and divided into 3 groups, and each group contained 375 pieces and divided to 5 subgroups, 75 pieces. In the first group, each of subgroup 75 pieces were detected in each of the concentrations of plant extracts. In the second group, almost 375 pieces. Each subgroup, 75 pieces, was detected in each of the concentrations of Protecto. And the third group, the same numbers in each of subgroups, or in each of concentration of Dorspan. Treated cardboard samples were placed in glass flasks, 2 liter. 50 pieces were used for each concentrations where termites were added, and the flasks were sealed using a piece of gauze to allow ventilations. Each flask contained 10 workers and 5 soldiers, almost total 15. With the same number, experiment was carried out with nephos, where each flask contained 5 nephos. Untreated control reflectors, 5 reflectors contained 10 workers and 5 soldiers, and 5 nephos, who were supplied with species of untreated cardboard paper only. Each concentration tested was reflected 5 times. Was reflected for 5 times. All the treatments were kept in darkness at 48 degree. The tests were halted. The number of surviving termites were counted, and the percent mortality. The laser concentrations 50 LC expressed as milligram per liter was calculated from log concentrations mortality registrations lines. In this table number 1, show gas chromatography, mass spectrometry, results the extract of lavender oil. We can see the high one second. Shows the result of this gas chromatography. The main consists where we can see the main consists of this oil. It's called linalool. Linalool 21.49%. We can see in oil, in lavender oil. In this extract of or milligrams oil, the main compound of the main consist of this oil is C-mol, the C-mol and the linalool. It's almost 14%. The table number 3, result of the extract of clove oil. We can see it's the main consist prestige in the oil. 99% is ethanol, called ethanol. The effect of tested plant extracts showed that the monogram extract has the highest toxicity against microterms of termites. After 7 days to this extract, the LC50 was 170 milligram in liter against soldiers and workers, and of course nephews. The lavender extract has less of toxic effect against the marjoram with an 50 LC of 1086 milligram per liter for the group of soldiers and workers and nurses. Clover extract showed the lowest insecticidal activity against the soldiers and workers. So we can see marjoram extract was most effective as insecticide against termites. Table number 4 shows that the value of those pen is for individuals and neighbors were 84 and less than 120 milligram per liter. The protector shows the value of LC50 less than 164 and 265 milligram per liter for protector. So we can see the effect, the huge effect, of course, of chemical pesticides compares of plant extracts, but also we can see the marjoram oil had the huge and the highest toxicity against termites. Table number 5 shows the effect of various compounds on the mortality of termites in laboratory test, in lab test. Our result shows that residual span caused the highest termite mortality of 80%, 80%, while protector ranked second. It's the second at 75% mortality. So the span causes the highest effect as we show. For the plant extract, marjoram caused the highest mortality at 70%. So we can see it's no difference or huge difference between chemical pesticides and plants extract and plants or the plant extracting. In this table, we can see here the mortality. Here is the mortality of protector. Here 80%. The second 75, and the plant extracts marjoram 70, lavender 50, clove is the finally is the weak effect of termites in 25%, almost 25%. Discussion, many plant extracts compounds can make a high mortality in termites. This is related to their mechanism of action on the insect nervous system. Plant compounds may also have neurotoxic modes of action. The data presented in this work clearly show a big difference in chemical components among plant samples depending on their origin, even in the same species. Chemical pesticides have the greatest impact on the mortality of the termites. As we show in the table or the numbers of percent. Due is a highly toxic of Dursban and the fact that it has been withdrawn for use in soil in countries that search for less toxic and natural termicides. Extraplants of is often the interest. We interested about plant extracts from plants show some promise as a plant-based toxicant for these needs. And this is all. So thank you for you listening, and any questions I will answer. Thank you.
Thank you. Thank you, Doctor, for your presentation. Is anyone having any questions? I hope so. No questions. Once again, thank you.
Thank you for your listening and for your waiting and also your time. Thank you. Thank you.
Thank you. Thank you so much.
Thank you so much.
Let's move on to our next session. The Dr. Iyad Soap from Lebanon University from Lebanon. Hello, Dr. Iyad. Are you ready with your presentation? Hello, Dr. Iyad. Can you hear me?
Hello, yes, now I can hear you.
Okay, so now are you ready with your presentation?
Yes, ready.
Okay, so now I'm going to make you the presenter.
Okay.
Yes, please click on the screen button to share your screen.
Okay.
Yes, your screen is visible to us.
Okay. And okay. Now it's
Yes.
visible?
Yes, you can present your stuff.
Okay. Good afternoon. Today I'll speak about the modulation of microRNAs as one molecular explanation of toxoplasma-associated brain cancer. I'm Iyad Subah. I've graduated from the Lebanese University having an M1 in molecular and cellular biology and a Master 2 degree in forensic biology, forensic sciences. Okay, here's the outline of my here's the outline of my presentation. Okay. First, I'll talk about the generalities about the Toxoplasma gondii, a parasite. It's from the class Coccidia and the order Eucoccidia. It causes toxoplasmosis in animal and a human by the infection by the cyst or the oocysts. It is an intracellular parasite. It belongs to the Apicomplexa phylum. It affects approximately 30% of the world's human population and it's the main cause of toxoplasmosis. Toxoplasma gondii is the etiologic agent of toxoplasmosis I mentioned before. It manifests as acute and the chronic forms. It infects the central nervous system and it is caused by the presence of fast replicating tachyzoids which deploy immune cells as vehicles to reach the brain and skeletal muscles where they convert into persistent bradyzoid cysts. For the life cycle of the Toxoplasma gondii, it has two definitive hosts, cats or any other felidae, and intermediate hosts as humans and warm-blooded animals. It has three forms: tachyzoids, bradyzoids, and sporozoids. Here, as this figure shows, the cat can ingest the three forms from any prey. First, we'll have the sexual reproduction or the gametogenesis. Micro and macrogametes will lead to will be zygote. Then, the rupture of the intestinal cells in the cat will cause the unsporulated or immature oocyst in feces in environment within 3 to 7 days, and the mature oocyst after 1 to 5 days being in the environment. Each sporulated oocyst contains two sporocytes, each which contain two sporozoites, or 2 to 4 sporozoites. And the ingestion by any warm-blooded animal causes an asexual life cycle to start over. This will reach to the stomach of the intermediate host, as humans, for example, and will release sporozoites. Sporozoites will change into tachyzoids in blood and invade any nucleated cell and can pass the placenta through the vertical transmission. And this will cause an acute infection. After 5 days, tachyzoids will transform into bradyzoids under immune response effect. Bradyzoids insist in the brain and skeletal muscle and can be reactivated when infected person become immunocompromised, as being for an HIV patient or after a transplantation, and this will be life-threatening. Intermediate hosts acquire infection through ingestion of sporulated oocysts or tissue cysts. Bradyzoids, they are slowly replicating, associated with chronic and latent infections, and the tachyzoid, they are rapidly replicating. Here are the figures showing the stages of Toxoplasma gondii as the sporozoites, the tachyzoids, and the bradyzoids with different types of microscopy and different magnifications. How it is transmitted to humans? No, wait. Okay, no. How it's transmitted to human? It's either by ingestion of the oocyst or the tissue cysts in undercooked meat, congenital or the transplacental transmission, or the vertical transmission by organ transplantation or by blood transfusion. Toxoplasmosis can be of two types, either the congenital toxoplasmosis or the adult toxoplasmosis. The congenital toxoplasmosis or by the placental transmission may cause spontaneous abortion, stillbirth, or even severe mental retardation and chorioretinitis upon birth. And the adult toxoplasmosis causes lymphadenopathy, ocular disease, neurotoxoplasmosis, and different psychiatric disorders related to toxoplasma. I'll go forward with the psychiatric disorders and what will happen in morphological changes in the okay. It will cause the brain inflammation, dilation in its ventricles, altered fiber density, loss of fiber continuity, reduction in the PSD95 and synaptophysin and synaptic proteins, moreover as in reduction in dendritic spines. Sorry, I'm facing some problems in changing the screen. Okay. As for these morphological changes, they will result in the neuropathies or how it will affect or toxoplasma will be associated with neuropathies disorders as depression and associated behavioral changes, schizophrenia, bipolar disorder, and the OCD or the obsessive compulsive disorder, as well as Alzheimer. The Toxoplasma gondii that affect the brain will affect the brain and any other part of the human system will cause modulation of different RNAs, especially the non-coding RNAs that are divided into two types: the shorter non-coding RNAs as microRNAs, short interfering RNAs, transfer RNAs, interacting RNAs, and small nucleolar RNAs, and the other type, which is the long non-coding RNA. It alters specifically the expression of microRNAs, crucial for the immune response in the host cell. The expression of 14% of microRNAs are altered following 24 hours of infection in a human foreskin fibroblasts that are under experiment. And in human macrophages, the upregulation of 8.3% and downregulation of 4.3% of the total microRNAs happened as the experiment showed. We have a specific type of microRNA clusters or groups that will be expressed and increase in expression with all strains infection as the microRNA 30C1, 125B2, and specifically we'll talk about it, the microRNA 1792 cluster. MicroRNA 1792 cluster is one of the upregulated microRNAs during infection. It inhibits the host cell apoptosis inhibits host cell apoptosis with a strategy while the ROC16 of the parasite is internalized into the host cell cytoplasm that will phosphorylate STAT3. The phosphorylated STAT3 binds to microRNA 1792 cluster binding site in the promoter region. Thus, this will upregulate its expression and will induce the bind to the 3' UTR of the BIM transcript. As we know, the BIM transcript or BIM transcript is the transcript related to the cell.
Hello, Dr. Hello, Dr. Iyad. Can you hear me? Hello? Hello, Dr. Iyad. Can you hear me? Hello? Hello, everyone. Due to some technical issues, Dr. Iyad has gone through power cut. So So let's move on to our next session by Dr. Ramdatta Bhatt from Tribhuvan University, Nepal. In the title of Production and Profitability of Hybrid Rice is influenced by different nutrients from management practices. Hello, Dr. Ram. Are you ready with your presentation? Hello, Dr. Ramdatta Bhatt. Hello, Dr. Ram. Can you hear me?
Hello.
Am I audible? Yes, doctor. Are you ready with your presentation?
Hello.
Hello, doctor. Can you hear me?
Yeah, I'm hearing.
Yes. Are you ready with your presentation?
Yeah, I'm ready.
Please click on the screen button to share your screen.
This screen is not shareable. Would you please help me?
Could you please? Could you please? Hello, Dr. Ram. We are hearing so much of disturbance.
Oh.
Yes, doctor. Please click
Yeah,
on
the
the.
screen is not.
Yes, please click on the screen share button to share your screen.
Okay, thank you.
Hello, Dr. Ram. Please share your screen.
It's not shareable. I'm trying. Would you please mark me as a presenter there?
Yes, I have already sent you the request for the presentation to share the screen. Just a second. Did you get
Oh,
the pop-up window?
yeah.
Sorry, doctor, we are hearing so much of disturbance from you.
Hello.
We are getting disturbance. Hello, Dr. Ram. Please disconnect your device from the internet. We have disturbance from your device. Hello, Dr. Ram. Can you hear me? Hello, Dr. Ram. Can you hear me?
Hello.
Yes, Doctor. Hello. Can you hear me?
Yeah, I'm listening.
Okay. So now I'm
If the
going to
is
make you the presenter.
okay, thank you.
Did you get the pop-up window? Please click on the screen share button to share your screen.
Yeah, I did not get that.
Now I'm going to make you the presenter. I already make you the
Okay, thank
presenter.
you.
Please click on the screen share button.
I did not get any kinds of such things.
Just a second.
No screens or cameras are being shared.
Did you get the pop-up window now? To share
Yeah,
your
I am
screen.
I am here via my smartphone iPhone. So is
It's
there
okay.
any different?
Yes. There is no problem. Please click on the screen share button.
I didn't get any kinds of such things, such marks.
Yes. Yes, we can able to see your screen.
Okay.
Okay. Yes. Please open your PPT file.
Okay. If you are seeing,
Yes. Yes. Please make it as a full screen. Huh? Okay.
here is a poster. If this is visible to all,
Yes, it is visible, but like your screen is some zoom out. We can't see the presentation clearly. Could you please rotate your screen so that we can able to see?
Is that visible?
Okay. Yes, it's visible. Yes.
Good afternoon. Good afternoon to everybody. I am Ramdatta Bhatta from Nepal, belonging from Tribhuvan University, Institute of Agriculture and Animal Science. Today I am going to present my presentation on the topic production and profitability of hybrid rice is influenced by different nutrient management practices. First of all, you can see your abstract. Here the government of Nepal has recommended blanket fertilizer application for rice cultivation, which results in lower nutrient use efficiency, particularly under rainfed conditions, with the aim of finding an appropriate nutrient management practices concerning rice production and profitability. A field experiment was conducted during rainy season of 2017 AD and 2018 AD at Kabri-Plansuk and Dang district of Nepal. Although 5 treatments compromising various nutrient management practices like as nutrient expert model, which is also considered as NE, use of leaf color chart, that is LCC, and government recommended fertilizer dose, GON, farm admin or FYM, and farmers field practice were laid out in RCBD with 4 applications in farmers field. The analysis of variance showed significant difference between treatment for test weight and grain yield in Kabri-Plansuk, whereas all traits except number of effective tillers were significant in Dang. This significantly higher grain yield and harvest index were obtained in nutrient export, followed by leaf color chart, and the overall store yield was highest in leaf color chart, followed by nutrient export in both the locations. Also, yield gap analysis suggested the nutrient export had 44.44% and 23.97% increase in yield as compared to farmer field practice in Kabri-Plansuk and Dang respectively. Both correlation study and biplot of principal component analysis signaled grain yield had positive correlation with all other traits. Furthermore, the net revenue was maximum for NE, followed by LCC in both the location. The benefit cost ratio was highest for NE, which was 1.55 in Kabri-Plansuk and 2.61 in Dang. On the basis of these findings, nutrient export and leaf color chart can be effectively used as nutrient management practices by the farmers to obtain maximum production and profitability in rice. And methods and materials. A field experiment was conducted in farmers field of Mahadevistan, one of the city of Nepal of Kabri-Plansuk district during rainy season of 2017, and in Lomai municipality, which is Dang district of Nepal. The site of Kabri is at 27 degrees 71 latitude north latitude and 87 degrees 61 second east longitude, which is 600 meter above sea level. An experiment site is located in a dry rain-fed region of Nepal with a subtropical climate consisting of wet summers and dry winters. The weather of the research area is presented in figure, but I do not have figure here. Field experiment was conducted Kabri and Dang district of Nepal during the Kharif season, that is rainy season of 2017 and 2018, with a hybrid rice transplanted to the main field after 22 days in the nursery bed as farmers practice. The today's experimental treatments were laid out on randomized RCBD design with 4 applications in both locations. Seedlings were transplanted 1 per hill with a spacing of 25 centimeter into 25 centimeter. That is, the seed rate was 10 kilogram per hectare. Three fertilizers named as urea, diammonium phosphate, and muriate of potash were used as a source of nitrogen, phosphorus, and potassium as per guidelines of Government of Nepal. In the nutrient export, fertilizer dose along the application time as per the recommendation of nutrient export model was applied. The model was run with the data of surveyed, interviewed farmer with questionnaire. In the leaf color chart, phosphorus and potassium was applied as per the recommendation made by Newton expert, and nitrogen was applied as per need on the basis of leaf color chart score. In the government of Nepal, recommended fertilizer dose, fertilizer in each plot was applied as per the recommendation by National Agriculture Research Council, that is one of the research institute of Nepal, that is 140 is to 60 is to 30 kg NPK per hectare. In the farm-aid manner, FYM was applied to each field at 15 ton per hectare. Measurement of the agromorphological traits such as plant height, number of effective tillers per meter square, length of panicle, test weight, stay and grain yield were recorded. The total cost and gross revenue of the rice production was calculated. The harvest index and fertility were taken, and economic parameters BC ratio were evaluated by using formula. Data analysis, MS Excel was used to enter and process all the data. And the combined analysis of two sites was carried out by Meta-R with the best linear unbiased estimator, that is blue. Minitab 14.0 was used to prepare the principal component biplot. Now, result and discussion part. Plant height and analysis of variance revealed a highly significant difference between the treatments for plants in Dang. But while in another district, Cabre, difference was not significant statistically. The overall mean of the plant height was 91.63 cm. The mean plant height was 84.47 cm and 95.78 cm in Cabre and Dang, respectively. Plant height was maximum in farmyard manner, that is 91.58 centimeter, and minimum in Newton export, that is 83.76 centimeter at Cabre district. While at Dang, maximum plant height was observed in Newton export, that is 99.89 centimeter, followed by government of Nepal, that is 98.41 centimeter, and minimum plant height was observed in farmer field practice, that is 88.3.39 respectively. And the number of effective tiller, a significant difference was found for the number of effective tillers per meter square at Cabre, while it was not significant at Dang. The mean number of tillers per meter square was 2,058,363, and in Cabre 315.50, and Dang and the overall field respectively. The number of effective tillers per meter square was highest in government of Nepal plots, that is 278.40, followed by leaf color chart, that is 273.00, and minimum in farmer field plots. While LCC had the maximum number of effective tillers per meter square, and Newton expert had the lowest number of effective tillers per meter square at harvest in Dang. Excuse me, am I audible?
Yes, doctor, we can hear you. Please continue.
Thank you. And next one is panicle length. Panicle length was statistically significant in Dang while non-significant in Cabre. The overall mean of panicle length was 24.76 cm. Cabre and Dang, both the districts had mean panicle length of 26.92 centimeter and 22.5 centimeter respectively. If you look at by district wise, panicle length in Cabre district, leaf color chart had the longest panicle length, that is 27.41 centimeter, followed by Newton export, that is 27.18 centimeter, and farm and manure and farmer field practice both had the shortest panicles length, that is 26.56 centimeter. If you look another district, that is Dang, the longest panicle length was found in Newton export, that is 26.31 centimeter, which was statistically superior to all other treatments. The shortest panicle length was found in farmer field practice, which was statistically at par with leaf color chart, government of nutrition, government recommendation dose, and farm aid minor. Fertility. If you look at fertility, in one of the district, Cabre, there was no significant difference between regards to percentage fertility, while in Dang, another district, there was significant difference. The overall mean of fertility was 77.40%. The mean percentage of fertility in Cabre and Dang were 72.6% and 82.2% respectively. The percentage of fertility ranged from 68.5% to 80.25%, with highest fertility recorded in LCC and lowest fertility recorded in FYM in Cabre. In Dang, one of the district among two, the percentage of fertility ranged from 67.24% to 90.52%. The highest fertility recorded in farm-aid manner was statistically superior to another treatments. It was followed by leaf color chart, that is 88.12%, and nutrient export, that is 87.56%. The lowest fertility recorded in farmer field practice. Similarly, test weight. If you talk about test weight, the mean 1000 grain weight was 15.7 gram in Cabre district and was 15.75 gram in Dang district. The overall mean was 15.73 gram. In Cabre, the maximum test weight recorded was 17 gram in nutrient export, which was followed by leaf color chart, that is 16.5 gram, while the minimum test weight recorded was 14 gram in farm-aid winner. Similarly, in Dang district, the maximum test weight recorded was 17 gram in farm-aid manner and leaf color chart, while the minimum test weight recorded was 14 gram in farmer field practice. Let's talk about grain yield. In one of the district, Cabre Palanchok, ANEVA elucidated significant effects of treatment on grain yield on rice at 0.05 level of significance. The highest grain yield was 5.84 ton per hectare in nutrient export model, followed by leaf color chart, that is 5.61 ton per hectare, and government recommendation dose, that is 5.34 ton per hectare, and similarly followed by FYM, that is 5.14 ton per hectare. The lowest yield was found in FYM, that is 4.10 ton per hectare. And similarly, the grain yield of Dang district, and here the highest grain yield of rice was recorded as 6.36 ton per hectare in nutrient export model, followed by leaf color chart, that is 6.17 ton per hectare, and followed by FYM, that is 5.8004 ton per hectare. And here the lowest yield was obtained in farmer field practice and government recommendation dose, which were 5.13 ton per hectare and 5.20 ton per hectare respectively, statistically at par with farm yield manner. Now, straw yield. There was significant difference between nutrient management treatments for straw yield in Cabre district and Dang district. The mean straw yield in Cabre was 10.9 ton per hectare and in Dang was 10.97 ton per hectare. The overall mean was found to be 10.93 ton per hectare. The highest straw yield was 12.04 ton per hectare in farmer field practice, followed by 11.50 ton per hectare in government recommendation, statistically at par with farmer field practice, while the lowest yield was found in leaf color chart, that is 9.50 ton per hectare in Cabre. Similarly, if you talk about Dang, the highest straw yield was found in leaf color chart, that is 12.87 ton per hectare, statistically at par with nutrient, which is 12.62 ton per hectare. The lowest store yield was found in farmer field practice, that is 12.04 ton per hectare. Now, harvest index. The mean harvest index was 33.92% and 35.76% in Cabri and Dang, respectively. If you talk about harvest index individually, in Cabre Palanchuk district, highest harvest index with 38.52%, followed by leaf color chart with 38.47%. The lowest harvest index was found in Farmel manner, that is 29.20%. In another distinct bank, significantly higher harvest index was obtained from nutrient export fertilizer recommendation than other treatments, followed by leaf color chart, which is 39.57%, which are statistically at par. The lowest harvest index found in FYM, which is 28.7%. Now discussion. This research was similar to one by where balanced use of fertilizer boosted plant growth and produced maximum height, whereas poorly fertilized plots produced plants with minimal height. The application of macronutrients according to site requirements leads to higher plant height in rice. Further, this finding is in line with who reported site-specific nutrient management had more effective tillers than farmer field practice. The reason for high store yield in leaf color chart and nutrient export model because of balanced nitrogen applied that influenced the vegetative growth, plant height, and number of tillers, leading to a high store yield. The nutritional management practices in Cabre and Dang district differed significantly in terms of grain yield. This finding is similar with one of findings which I cited here, and the evidence of nutrient exports to over government recommendation of recorded uses nutrient export maize and wheat. Hence, nutrient export is far better nutrient management practice over government recommendation dose and farmer field practice. The grain yield is a complex state attributed by different components, which found higher yield in nutrient export is the result of better panicle length, number of effective tillers per square meter, and test weight, which also result in higher harvest index and 30% less fertilizer use. Combined analysis of store yield and harvest index in Cabre district, the nutrient management and location interaction showed significant effect in the combined store yield and harvest index across both the location, while treatment effect was found to be statistically non-significant as here. In terms of store yield, the highest yield was given by any nutrient export and lowest yield by government recommendation dose. Now, yield gap analysis. Yield gap at Cabre. In comparison with farmer field practice, yields from nutrient export increased by 42.4%, followed by leaf color chart and government recommendation dose by 36.82% and 30.24% respectively. Similar results of grain yield increased by 37.62% in nutrient export rice recommendation over the farmer field practice. These results are in agreement in wheat reported nutrient export produced 57% more grain yield than farmer field practice. This implies that in contrast with farmer field practice, different treatments showed increased grain yield. correlation among different rice states across both the location. The correlation coefficient between different rice state and harvest state index is presented here. The grain yield had significant strong positive correlation with fertility 0.90, test weight 0.93, stock yield 0.89, and harvest stock 0.94. There was significant strong positive correlation of harvest index with test weight and significant positive correlation between grain yield, panicle length, and stock yield. Positive correlation between grain yield, panicle length is in the line with positive correlation with here was supported by finding similarly positive correlation of grain yield with tiller weight and panicle length also reported a positive relation of plant height and number of tillers per plant with grain yield of rice. Now, conclusion. Nepal's rice production is much below its potential due to farmers' imbalanced fertilizer application practices. The proper nutrient management, that is the correct amount of timing of fertilizer application, minimizes the yield gap in crops. Nutrient export and leaf color chart have both been proven in increasing grain yield and stock yield. Furthermore, the highest benefit cost ratio for rice production was achieved in nutrient export due to its high net revenue for farmers. According to BLUE estimates, the straw yield and harvest index were both affected by nutrient management and location interaction. Furthermore, all the tested traits were found positively correlated with the grain yield of rice. This experiment concluded that nutrient management practices are most appropriate for increasing the profitability of farmers producing rice. Yeah, thank you very much for listening. Thank you very much. Hello.
Thank you, Dr. Ram, for your presence and presentation. Any questions? So I hope no questions, Dr. Ram. Once again, thank you for your valuable presentation.
You are welcome. Thank you.
Thank you. So by this, we came to the end of day 2 webinar, and I would like to thank you all the attendees and participants of today's webinar. Hope you enjoyed the webinar and gained some knowledge from the sessions, and we request you to attend to. If you have any queries, you can email, and we will be very glad if you could join us in the next webinar, which is scheduled, and the dates will be informed to you by email. We will be very happy if you could share your opinion and feedback with us. Thank you for your attention. Have a nice day.